Genetic analysis methods: polymerase chain reaction, PCR. Polymerase chain reaction, PCR, is a revolutionary genetic analysis technique that amplifies specific segments of DNA to make them easier to analyze. This method has wide applications in genetic testing, including in pre-implantation genetic testing, PGT, forensic analysis, disease diagnosis, and research. PCR enables the identification of genetic mutations or variations in a targeted DNA region, even when the DNA sample is very small. In the context of pre-implantation genetic testing, PGT, PCR is commonly used to detect single gene disorders, such as cystic fibrosis, sickle cell anemia, or Huntington's disease, and to analyze embryos for genetic disorders before they are implanted during IVF. This section will explore the principles of PCR, its applications, advantages, and limitations, particularly in the context of genetic analysis in PGT. 1. Overview of polymerase chain reaction, PCR. PCR is a method used to amplify a specific segment of DNA in a test tube, allowing researchers to produce millions of copies of a particular DNA sequence. This process is critical in genetic testing because it allows for the analysis of small or degraded DNA samples. Steps of PCR: denaturation. The DNA sample is heated to around 94 to 98 degrees Celsius to break the hydrogen bonds between the two DNA strands, creating single-stranded DNA. Annealing. The temperature is lowered to around 50 to 65 degrees Celsius so that short DNA primers can bind to the specific regions of interest on the single-stranded DNA. Extension/elongation. The temperature is then raised to around 75 to 80 degrees Celsius, and the DNA polymerase enzyme extends the primers to synthesize new strands of DNA that are complementary to the original DNA template. Amplification. These steps are repeated for 20 to 40 cycles, each cycle doubling the amount of the specific DNA target, resulting in millions of copies of the target DNA sequence. Key components of PCR: DNA template, the sample DNA that contains the region of interest. Primers, short sequences of nucleotides that guide the polymerase to the target region. DNA polymerase, an enzyme that synthesizes new DNA strands. Nucleotides, dNTPS, the building blocks that make up the new DNA strands. Buffer solution, maintains the optimal environment for the enzyme activity. 2. Applications of PCR in genetic analysis. In PGT, PCR is commonly used to detect genetic mutations or single gene disorders in embryos, as well as to confirm the presence of certain genetic conditions that may be passed on from one or both parents. A. Detection of single gene disorders. PCR is highly effective for detecting single gene disorders that are caused by mutations in specific genes. Some examples include cystic fibrosis, mutation in the CFTR gene; sickle cell anemia, mutation in the HBB gene; Huntington's disease, mutation in the HTT gene; Duchenne muscular dystrophy, mutation in the DMD gene; pre-implantation genetic testing for monogenic disorders, PGTM. PGTM is used to test embryos for specific genetic disorders inherited from one or both parents. In PGT-M, PCR is used to amplify DNA from embryo biopsies to test for monogenic diseases before the embryo is implanted during IVF. C. Detection of embryo anomalies. PCR can also be used in embryo screening to detect specific genetic mutations in embryos, allowing clinicians to choose genetically healthy embryos for implantation. D. Gender selection. PCR is sometimes used for gender selection in cases of X-linked disorders. By identifying the presence of Y chromosome sequences in the embryo, gender can be determined before implantation. Third, advantages of polymerase. Chain reaction, PCR. PCR has transformed genetic testing and provides many advantages in both research and clinical applications. A. Sensitivity. PCR can amplify very small amounts of DNA, allowing for genetic testing even when only a small sample is available, such as in the case of embryo biopsies. B. Speed. PCR is a relatively fast process, and results can be obtained in hours compared to other methods like genetic sequencing, which can take much longer. C. Specificity. PCR can be designed to amplify specific regions of DNA, making it an ideal tool for targeted genetic testing. It can detect specific mutations or genetic disorders with high precision. D. Cost effectiveness. PCR is one of the more cost-effective methods for genetic testing compared to next-generation sequencing, NGS, or whole genome sequencing, making it accessible for widespread use in clinical laboratories and fertility clinics. E. Multiplexing capability. PCR allows for the amplification of multiple genes or targets simultaneously in a single test, known as multiplex PCR, which can be highly efficient for screening for several genetic conditions at once. Fourth, limitations of polymerase chain reaction, PCR. While PCR is a powerful tool, it does have certain limitations that should be considered when using it for genetic analysis. A. Detection limitations. PCR is excellent for detecting specific mutations, but may not be suitable for detecting complex genetic abnormalities such as large chromosomal abnormalities, deletions, duplications, or structural rearrangements. B. Risk of contamination. PCR is highly sensitive, so it is susceptible to contamination. Even trace amounts of foreign DNA in the lab can lead to false positives, which is why proper laboratory techniques are essential. C. Limited coverage. PCR can only detect the regions of DNA that are targeted by the primers. It cannot provide a comprehensive overview of the entire genome, which limits its use for whole genome analysis or detecting multiple mutations across a gene. D. Amplification errors. PCR can be prone to errors during the amplification process, such as misincorporation of nucleotides or primer dimer formation, which may result in false negatives or false positives. E. Not suitable for all disorders. PCR is typically used for single gene disorders and may not be suitable for disorders caused by multiple genes or epigenetic modifications. 5. Conclusion. Polymerase chain reaction PCR is an essential tool in genetic analysis, particularly in pre-implantation genetic testing, PGT, for detecting single gene disorders and embryo screening in IVF. It offers high sensitivity, speed, and specificity, making it ideal for genetic testing of small samples, such as those obtained from embryo biopsies. However, it has limitations in terms of coverage and its ability to detect large-scale chromosomal abnormalities or complex genetic disorders. Despite these limitations, PCR remains a valuable technique in the detection and prevention of genetic diseases.